the first test that we are going to be talking about for the Unit 2, identifying gram-positive cocci bacteria, is going to be PEA, which is phenylethyl alcohol, Agar. PEA is an agar plate, and it is a selective medium because it restricts the growth of gram-negative organisms. Because phenylethyl alcohol is going to disrupt the gram-negative cell wall, which is going to cause the gram-negatives to become leaky. And one thing in particular, which is cellular potassium, is going to leak out of the cell, and that has been linked to disrupting DNA replication in gram-negative organisms. So this media is selecting for gram-positives and selecting against gram-negatives. Now, it doesn't completely prevent all gram-negative organisms from growing at all, but it greatly reduces the amount of growth that you may see. So as you can see on this photo, organism A on that PEA plate is going to be gram-positive. It grew very, very well. Whereas bacteria B did not grow quite so well, and that's because DNA replication was disrupted in this organism. And bacteria C, which is also gram-negative, had an even worse time growing because it was just more susceptible to the phenylethyl alcohol. So what you see on this plate is a technique called spot inoculations, where you'll get maybe some broth or just a sample of the bacteria that you're testing on your inoculation loop, and you just literally set it down on the agar in one spot, and then you just draw a little line. So you put the bacteria in one spot and then streak it down just a little bit to form that little line. Because the goal of this plate is not to get isolated colonies or anything like that. This is strictly to see, is this a gram-positive organism or is it gram-negative? Because sometimes when you're out in the field, right, or even just in a lab, gram staining can be difficult. It's a very picky stain. And so you might be like, well, I think that this is gram-positive, but let me confirm. So this is a really good confirmation media because you can do a very quick spot inoculation. It takes about 20 seconds. And you can look at it the next day and be like, okay, this grew really well. It definitely is gram-positive. Or you'll be like, ooh, this thing didn't grow at all. I was wrong. This is actually a gram-negative organism. Okay, so it doesn't really help you identify your unknown, but it can confirm whether or not it is gram-positive because it is selective for gram-positive organisms. So here are the results from the PEA lab. And so for this lab, it was a very straightforward lab. There was just one PEA plate and one TSA plate, which is a regular agar plate. And as you can see, it was divided into 3 sections. And you can either spot inoculate or you can do a little streak like this. Again, the goal is not isolated colonies, but we wanted to illustrate how well some organisms did or did not grow. So on a TSA plate, again, this is a general media, so anything should grow just fine. And so as you can see, all 3 organisms grew, and they grew perfectly fine. Remember, y'all, organism B is Pseudomonas aeruginosa, and it has a natural greenish blue pigment, and so that's why it looks the way it does on the TSA plate. That is completely normal. But on our PEA plate, which is selective medium, notice that Staphylococcus aureus, our gram-positive organism, definitely grew the best. Okay, E. coli, which is a gram-negative pathogen, didn't grow at all. And Pseudomonas was able to grow just a little bit, okay, but it's gram-negative, so its DNA synthesis was greatly disrupted, which is why we got these results. All right, the next test that we're going to talk about is the Catalase Test. And the Catalase test is used to differentiate the two big groups, our Staphylococci and our Streptococci. And so we've talked a lot in lecture already about the enzyme catalase. Remember, it breaks down hydrogen peroxide in order to form gaseous oxygen and water. And so it's that gaseous oxygen that produces the bubbles that you sometimes see if you've ever tried to clean out a minor flesh wound with peroxide. And so remember, that's why peroxide isn't the most effective at minor skin wounds, because a lot of the bacteria on the skin or right below the surface of the skin have the enzyme catalase. But peroxide is really good on inanimate objects. So to do this test in the lab, you just use a microscope slide. You'll put a little bit of your cells on the slide, and then you will drop hydrogen peroxide onto it. So it's a very cheap, easy test, and you get immediate results. Either it bubbles, like you see on the left, or it does not bubble, which you see on the right. So bubbles is a positive result for the catalase test, and no bubbles is a negative test. That means the organism does not have catalase. So I want to take a second to remind you of what an observation is versus a result versus a conclusion, because this is going to be important on the lab exam and on your unknown report. An observation is what you see. So for the catalase test, your two options for observations are bubbles and no bubbles. Okay, so what do you see? You add the peroxide and bam, there's bubbles or there's not. A result is always positive or negative. So if your observation was bubbles, your result is positive. If your observation was no bubbles, your result is negative. Your conclusion or interpretation is the so what that you're trying to answer. Well, what do bubbles mean? Or what does a lack of bubbles mean? And so the conclusion or interpretation would be, if your observation was bubbles and it was a positive result, your interpretation is the bacteria has the enzyme catalase. If your observation was no bubbles, your result was negative. Your conclusion or interpretation is that the bacteria does not have the enzyme catalase. So notice on the PowerPoint, lots of information that you need to know, but pay attention to where we indicate what's going to grow and what's not, okay? Because all Staphylococci are going to produce bubbles. All Streptococci are not going to produce bubbles, okay? And we'll come back to that flow chart in a minute, but this is information that you need to be filling out your flow chart each week. So before we get to the flow chart, this is just kind of a reminder about peroxide and catalase. So Staphylococcus do have some defense mechanisms against hydrogen peroxide. However, peroxide is still effective towards them. They're just, Staphylococci tend to be pretty challenging to kill sometimes. They're not quite endospore level, but they can still be a challenge. So if the bacteria has the enzyme catalase, it can actually protect those bacterial cells from any peroxide that is produced naturally as a result of some of the chemical reactions inside of the cell. Because if peroxide is made as a byproduct of a chemical reaction in the cell, they can then use the enzyme catalase to immediately break down that dangerous hydrogen peroxide into water and oxygen. Okay, so that's why bacteria that have catalase are automatically given this protection from it when we use peroxide as an antisepsis. Okay, but if you have a really highly concentrated hydrogen peroxide and you drown them in peroxide, then it can still be effective. Okay, but again, hydrogen peroxide is better on inanimate surfaces than flesh wounds. All right, so some Staphylococcus are going to, like Staphylococcus aureus, is going to have a pigment, and it turns out that the yellow pigment made by some strains of Staphylococcus aureus actually helps protect it from the effects of hydrogen peroxide. So we can actually say that the pigment is going to be a virulence factor, which is pretty snazzy. Okay, so what should you be taking away from these PowerPoints in the handouts? Okay, so of course you want to be answering the questions on the handouts. I will do my best to make sure that I'm addressing those questions in my little spiel. So make sure you're answering those, but you also want to be filling out your flow chart. Okay, so this is just a blank one because it's easier for me to show you guys these. So let's go ahead and fill it out. So far we've only talked about the Catalase test, which is actually the first step in identifying your unknown. Because remember that PEA plate just says if it's gram-positive or not. And both our Staphylococci and our Streptococci are gram-positive. So when we're actually trying to figure out the unknown organism that you have, catalase is where you start. So you do the Catalase test and you have two options, positive or negative. So if there are bubbles, then your result is positive and you know you're dealing with a Staphylococci. If on the other hand, the results are negative, there are no bubbles, then you are dealing with a Streptococci organism. Okay, and so both this blank copy and a filled out version are available on Blackboard. And so as we go through things, you're going to want to be filling out that chart based on the information provided. Okay, it doesn't give you all the answers. You have to use your resources to figure out what goes in the rest of these blanks. Okay, all right, let's do one more test on this video. Excuse me, and that is going to be mannitol salt agar. This is an agar plate, as you'll see a picture of it on the next slide. And we call it MSA because it's just shorter and easier to say. But knowing what MSA stands for is very helpful because MSA is both a selective and differential media. And that is because the salt, which is the S in MSA, salt is a selective ingredient, right? Only salt-tolerant organisms can grow in the presence of salt, okay? So if you see any type of growth on this plate, you know it's positive for being salt tolerant. If there's no growth on the plate, then the organism could not handle the salt. So they are considered to be salt intolerant. So then if an organism can grow on the plate, you know it's salt tolerant, but now we can also differentiate our salt tolerant organisms because our Staphylococci, some of them are salt tolerant. You find them on our skin, right, where it's sweaty sometimes, which is salty. Okay, so some organisms can survive the salt. So we can differentiate some of the Staphylococci based on their ability to ferment mannitol. Mannitol is a sugar. That's the M in MSA. So if an organism can ferment the sugar mannitol, then the pH is going to decrease. Because remember, I'm going to say this 100 times, fermentation naturally produces acidic byproducts. So the pH is always going to decrease if fermentation occurs. There is a pH indicator in MSA plates, and that pH indicator is called phenol red. So in the presence of fermentation, or I guess if fermentation has occurred, that pH is acidic, and phenol red turns yellow, a very vibrant yellow. However, if fermentation did not occur, then the pH doesn't change, and it remains its natural reddish color in the media. Okay, so again, we have two possible results, positive for mannitol fermentation or negative for mannitol fermentation. So let's take a look. So if you streak your unknown organism on an MSA plate and there is no growth, then you know it is salt intolerant. And if nothing could grow on the plate, then we have no clue about its ability to ferment mannitol because it couldn't grow. Nothing is there, okay? So Streptococcus pyogenes and other streptococci species, which are all salt intolerant, would give you this result because they are killed by the presence of salt in the media. However, as I mentioned before, our staphylococci are salt tolerant, so you are going to get growth on the plate. But again, now that something has grown, we want to differentiate them, right? We're trying to be able to differentiate our staphylococci species. So it turns out that Staphylococcus epidermidis produces colonies on the plate, and the plate stays the same color, okay? So let me go back a second. This is the normal color of the plate, right? The organism didn't grow, there was no fermentation, there was no anything. So this is the natural color of an MSA plate. And if you look closely, it got a little bit more pinkishy, right? But it's still in the red family. And so we say that no fermentation occurred because, well, quite frankly, it's not yellow. Your options here are yellow or not yellow, okay? So it's still red or reddish color, and so you know that the pH did not decrease, so it is negative for fermentation. And so Staphylococcus epidermidis gives you this result. Now, if our unknown is the dangerous pathogen Staphylococcus aureus, this organism can ferment mannitol, so it is going to turn the media bright yellow. Okay, so we know that it is positive for being able to be salt tolerant, and it is positive for mannitol fermentation. Okay, so notice though that there is one member of the Streptococci family that happens to be salt tolerant, which is why one of the reasons why it's no longer in the genus Streptococcus, like the rest of them are, because this guy was kind of an outlier. And as we learned more about the Enterococcus genus, we eventually moved this organism. So Staphylococcus aureus and Enterococcus faecalis would give this result. Okay, so let's just do a side-by-side comparison. So you can see on the left-hand picture that there was no growth at all, which is good because that's a Streptococcus organism. They are salt intolerant. But you can see the streptococci that is able to grow Enterococcus faecalis. So you can clearly see growth versus no growth, but you can also see that yellow color change that happens when fermentation occurs versus no fermentation. And you see that again on the right. Here we have two salt-tolerant organisms, which means they belong to Staphylococci. And one could ferment mannitol, which you see on the right. It's a yellow color change. So that's Staphylococcus aureus. And on the left would be Staphylococcus epidermidis. Okay? All right, so make sure you're filling out your flowchart and your handout. And as always, let me know if you have any questions.